Welcome, everyone. Really nice to hear that we have so many attendees for this webinar. I'll just briefly introduce that. We do a webinar on vitamin D immunity and COVID-19, which will be presented by Martin Hewison, who's already here. Martin is a collaborator from Birmingham. I put this abstract up so you could still read it if you hadn't already, but that was online too, and probably the reason most of you joined. Just briefly, there we go. The webinar is organized by 3 organizations together, and then it's probably nice to introduce these organizations to all of you too, since most of you are probably a member of just 1 or 2 of these. NUGO is the nutrigenomics organization, and is nowadays a foundation. One started as an FP6 project or network of excellence, actually still is. So a lot of nutrigenomics researchers are still part of that organization and try to build up that research field. Of course, like every research field, but especially in these health fields, that includes a lot of data and data analysis. And there is a connection between that same nutrition research community and ELIXIR, where ELIXIR, since ELIXIR actually is the European Bioinformatics Research Infrastructure ESFRI program. And it now has its own community for food and nutrition. Finally, this is not just about nutrients and vitamin D, it's also about COVID-19, and there is an, I think, very interesting project in which we try to describe the biological and molecular mechanisms behind COVID-19, which is the COVID-19 disease map project. And I hope we really have people from all these 3 organizations on board now. This is something different, but this was the direct reason that I started to collaborate with Martin Hewison. We had a collaborative PhD student not very long ago that actually looked at vitamin D and immunometabolic effects of vitamin D on the integrated system. It was actually also a really nice drive for a direct one-on-one collaboration between a Dutch and a UK university, which will probably after Brexit become more important. And this finally is one of the ways I learned to think about all these measures about COVID-19, because I anticipate one of the questions people will have is, yeah, well, we're talking vitamin D, how important is that even? And isn't that less important than one of the other things? This is a drawing that was inspired by original work from Ian McKay from Australia, who is a virologist and actually very active on Twitter. And I happen to know him before all this burned loose. And this drawing actually nicely slides how every single measure actually adds to protection against infectious diseases, although none of them are perfect. There's actually this nice mouse in there that's eating part of the cheese and lowering the protection, which is what the role of misinformation basically is. And with that, I'll give the word to Martin.
Thank you, Chris, for that kind introduction and setting the scene, as it were, for my presentation today. I'm just going to share the screen now. Good, well, afternoon, everyone, again. I'd like to begin by thanking Chris, firstly, for inviting me to give this presentation, and then also to NUGO, ELIXIR, and the COVID MAP project for organizing this webinar, and in particular, Xenia and Erin for organizing the meeting itself. The topic of my presentation today is one that's really been in the public eye in the last few months. It's interesting, I was at a meeting at the weekend where somebody pointed out that since lockdown in the UK started in March last year, there have been almost 2 manuscripts per day published on vitamin D and COVID-19. So there's an awful lot of stuff out there. And And what I'd like to do in the next 30 minutes or so is really just try and provide some clarity and context to this story. And I'd like to do this in two ways. Really, firstly, by emphasizing the key issues that I feel that are associated with the story. And the first is that the prevalence of vitamin D deficiency around the world, but particularly in countries like the UK. And the second, I think, is the most important one, is that this idea, this rationale for vitamin D and its link with COVID-19 really stems from basic fundamental understanding of the mechanisms that link vitamin D and the immune system. And I think that's a message that sometimes gets missed in many of the other stories about vitamin D and COVID-19. Okay, I'll begin by saying I have no conflicts of interest to declare. As Chris pointed out, I work at the University of Birmingham, which is in the very center of England. And really for the last few centuries, England has been at the very center of the vitamin D story. This really started in the 17th century when two English scientists, Francis Gliss and Daniel Whistler, provided the first description of vitamin D deficient rickets. In fact, actually, Daniel Whistler was working at Leiden University at the time, so it wasn't entirely in England. But nevertheless, in the centuries after that, really, England became the center of the rickets world, to the extent that actually rickets became known for a while as the English disease. And this was really because we were also at the center of the development of the Industrial Revolution. And we have these very sad pictures here of young children working in mills, factories, and generally not being in very good environments or very vitamin D friendly environments, to the extent that rickets became a very common feature in England. And the sad thing is that rickets story really hasn't gone away. If you go to any pediatric clinic in the UK, there's still a relatively high level of rickets reported in some communities. And I think this is a key issue that's really attached to the story. But nevertheless, in the last few years, what has really attracted people's attention in relation to vitamin D has not necessarily been the rickets story, but the link between vitamin D and other health issues for humans. And in particularly in the last few months, the potential link between vitamin D and COVID-19. And I'll come back to that in the rest of my talk. Nevertheless, I think although England's the center of vitamin D deficiency, as far as I can see, we tend to have a very conservative attitude to the vitamin D story. The government recognizes that we're all at risk of vitamin D deficiency, but the message tends to be keep calm and take vitamin D, but make sure that it's the lowest dose available. Don't go overboard too much. So for the rest of my talk today, I'd really like to focus on 4 key areas that I think are important. The first is just to give you some idea of how what vitamin D is and how it works. And secondly, to give you some idea about why we might be interested in vitamin D and the immune system per se in general. And then also, why specifically has vitamin D been linked to COVID-19? And then I'd like to end with, what do we do next with vitamin D, particularly in relation to COVID-19? And there, I think maybe some of that may come out in questions that we have after the talk. So to begin with, what is vitamin D and how does it work? Okay, well, this picture shows here that actually in reality, even in northern countries and northern Europe, you're still going to get most of your vitamin D from the action of sunlight on your skin to produce vitamin D3. Some countries will have some intake of vitamin D from the diet. In Scandinavia, for example, they tend to eat more fatty fishes like mackerel, herrings, and so on. So you may get a little bit of vitamin D from the diet, but in general, sunlight is your main source of vitamin D. But that vitamin D you produce initially has to undergo several metabolic steps to become active. The first is in the liver to produce 25-hydroxy vitamin D, or 25D for short. This is the main circulating form of vitamin D, and it's the one that we tend to measure when we're talking about vitamin D status. So if you're vitamin D deficient, it's because your 25D levels are low. But this isn't an active form of vitamin D, it's inactive. And to produce active vitamin D, we have to have a further metabolic step, which takes place primarily in the kidneys via the enzyme CYP27B1 to produce 1,25-dihydroxy vitamin D, or 125D for short. And this is the active hormonal form of vitamin D, and acts like all the other steroid hormones, that it interacts with a nuclear vitamin D receptor within target tissues, shown here as skeleton and the gastrointestinal tract. So this represents the endocrine hormonal arm of vitamin D's functions, and it's the one we know that is primarily associated with its effects on bone and calcium homeostasis. So this is the pathway that's really involved in protecting against rickets. But in the last 10 years or so, we've become aware that there is another way that vitamin D can also function. And this is because in some target tissues, in addition to having the vitamin D receptor there to respond to 125D, these tissues also express the vitamin D activating enzyme. So they're able to take this precursor circulating form of vitamin D, 25D, and convert it within those tissues to 125D. So you have this endogenous effect. And this is known as an intracrine mode of action. And it's very common in tissues such as the placenta, shown here, the spleen representing the immune system. And of course, there's been a lot of interest in how this mechanism is important within lung tissues in relation to COVID-19. And what's important about this mechanism is it appears to be far more dependent upon the availability of 25-hydroxyvitamin D than the endocrine arm of vitamin D's actions. So this part of vitamin D function could be far more variable depending on your vitamin D levels. If you're vitamin D deficient, it's possible that these actions will become severely compromised. And that's important when we start to look at the levels of vitamin D or 25D that are present in countries like the UK. So this is a publication from my colleague Suma Uday at the University of Birmingham, along with Wolfgang Högler, who showed that the levels of vitamin D in newborn children are extremely low. So this was using heel print blood spots taken from children just after they're born. And these samples were taken during the winter months and during summer. We have two sort of lines here, one showing if you're above that line, your vitamin D is sufficient. If you're below this lower line, you're vitamin D deficient. And what we see here is that in the winter months, nearly all the children that were born to UK mothers, in this study, it was 3,000 children. And you can see that they're all just about on this line or below the line, showing that all children appeared to be in danger of being vitamin D deficient when they were born during the winter months. This was particularly true of children from mothers who had a darker skin pigmentation. So if you were Black or Asian, severe vitamin D deficiency in these children. It got a little bit better in the summer, but in general, the levels of vitamin D in this population are very low. And this is worrying because within the UK, this is another publication published by Suma and Wolfgang a couple of years prior to this, where they showed that adherence or compliance with vitamin D supplementation advice is extremely low in the UK. If you look here in France, 90% of children in their first year take vitamin D supplements. In the Netherlands, it's even better. It's 90 to 95%, whereas in the UK, it's 5 to 20%. This is the lowest in the whole of Europe. So there's a very poor understanding of the problem of vitamin D deficiency in countries like the UK. So of course that has a particular impact on diseases such as rickets. But why are we interested in this in relation to other health effects such as the immune function? And really that's because when we look at vitamin D, 25D deficiency, of course, it has an impact on rickets, osteomalacia in adults, and other bone diseases such as osteoporosis. But in the last 10 years, there's been this increasing awareness of how vitamin D deficiency is also linked to other human health issues. And this is particularly true of immune-related disorders, infectious diseases such as TB and leprosy, but also autoimmune diseases, multiple sclerosis and type 1 diabetes have all been linked or associated with vitamin D deficiency. And in the last few years, there's been particular interest in how vitamin D deficiency could be linked to acute respiratory infections such as common cold, influenza, and in the last year, COVID-19. However, that's not really the reason that we become interested in vitamin D in the immune system. Really, we've got to go back to the 1980s when really some fundamental observations were made about the vitamin D system in the immune system. The first of these observations was that almost all the cells from the immune system express the vitamin D receptor, the one that responds to active vitamin D. And in addition to that, cells from the innate immune system, such as macrophages, the dendritic cells, also express the vitamin D activating enzyme CYP27B1. So these cells can take inactive vitamin D, activate it, and use that within the local immune environment. And perhaps the best example of this is studies that we did when I was at UCLA with Robert Modlin, which looked at how macrophages respond to pathogens such as Mycobacterium tuberculosis. And one of the key observations that came out of this was that the pathogen in this case was phagocytosed and entered cells. But in addition to that, it also signaled via toll-like receptors to trigger a number of genes being regulated. And prominent amongst these genes was the vitamin D activating enzyme and the vitamin D receptor VDR. And so this is a very simple observation, but what we were able to show that was if we increased vitamin D supplementation and raised the serum levels of 25 hydroxyvitamin D, this then entered the macrophages, could be converted locally to 125D, and this in turn could interact with the vitamin D receptor and trigger regulation of gene expression. And amongst those genes that we were able to show were regulated were antibacterial proteins such as cathelicidin, which then improved the killing of the mycobacterium. So this is a very simple model which said if you increase vitamin D supplementation, you get increased bacterial killing. And it's a very fundamental model that's been applied to many other aspects of vitamin D and immune function. Since then, we've expanded this model to include other antibacterial effects. So it's not just cathelicidin that's induced. Beta defensin 4 is also beta defensin 2 is increased. Vitamin D enhances autophagy in cells, so it improves the environment for bacterial killing. And also vitamin D regulates the levels of intracellular iron that are in cells like macrophages. So it limits the amount of iron that's available for pathogens to use as a fuel. So on multiple levels, vitamin D can have very potent antibacterial effects. And what we've realized in the last year or so is that many of these mechanisms can then be linked to antiviral responses for vitamin D. So this is a rather complex picture, but it essentially shows that the same autocrine mechanism in which you have production of active 125D locally within macrophages interacting with the vitamin D receptor can then trigger a number of antiviral mechanisms, including, again, the induction of autophagy, which can help with viral clearance, and some of these other intracellular factors that are known to help limit viral reproduction. So again, we have the same fundamental concept, was if we vitamin D supplement, we raise serum 25D and improve viral clearance. So that's within the innate immune system, a very simple model that involves the local production of 125D. But of course, that local 125D production within the innate immune system doesn't just have to stay within the innate immune system. There are vitamin D receptors present on many cells outside of that and within the adaptive immune system. So the 125D produced here can impact the adaptive immune system as well. And the basic model we now have for this is that, yes, cells such as dendritic cells are able to use their vitamin D activating enzyme to produce 125 dihydroxyvitamin D locally. That could act on the dendritic cells themselves. That's certainly one possibility. But that 125D can also be released to interact with T cells. And the predominant action of vitamin D in this setting is anti-inflammatory. It acts to suppress Th1, Th17, and T follicular helper cells to provide a strong anti-inflammatory signal, whilst at the same time promoting the development of regulatory T cells or Tregs, so that you have an improved tolerogenic or pro-regulatory response. So it's doing double duty, as it were, within the adaptive immune system. So we like to present vitamin D as being something that can be a double-edged sword for immune function. Now, normally a double-edged sword means that one side's good and the other side's bad, but in this case, both sides are really good. You have a strong anti-infection effect of vitamin D within the innate immune system, but you also have a strong regulatory or tolerogenic effect in suppressing inflammation and preventing tissue damage. So with that basic idea of how vitamin D acts on the immune system, how has this then been linked to COVID-19? Well, as I mentioned a few slides ago, of course, there have been studies that have shown that low serum 25D levels can be linked to increased risk of COVID-19. And of course, this is just a link, it's just association. It doesn't prove causation. But there have been some other studies that have shed a little bit more light on this. And some of these actually occurred before the current pandemic. So if we go back to 2017, Adrian Martineau and his team at Queen Mary University in London produced a very nice meta-analysis of 25 studies that looked at the impact of vitamin D supplementation. So this is giving vitamin D and how that prevented episodes of acute respiratory infection, that's colds and flu, in the recipients. And what they found was that in general, most of these studies were positive. They had a positive effect on and preventing at least one acute respiratory infection in the year during which the supplementation was taking place. So in fact, there's a significant effect with an odds ratio of 0.8, but actually this effect was improved if you just focused on those individuals who are vitamin D deficient to start with. So the supplementation didn't really work so well in people who are already vitamin D sufficient. And that's a key point to make. We should always be looking for people who are vitamin D deficient when we think about vitamin D supplementation. And the other key point this study brought out was that vitamin D supplementation works best if you use less and more often. So daily or weekly dosing worked really well, but big bolus doses given one-offs or once a month did not work so well. These are really important points to bear in mind when we look at COVID-19. And what this study also showed, I think, is that we have to remember that when we're looking at the action of vitamin D on immune function or on COVID-19, there are really two sides to the story, and it's not the two sides to the sword that we have here. The first is that when we look at the antiviral or antibacterial effects of vitamin D, we're talking about a community public health issue here, where we're using daily or weekly low doses of vitamin D supplementation to essentially limit infection by a pathogen, a bacteria or a virus. By contrast, when we look at the anti-inflammatory effects of vitamin D, here we're looking at effects of vitamin D that could be achieved for patients who've already been hospitalized. So here you've got to have something that works quickly, and it tends to be using bolus doses of high levels of vitamin D, or by using alternative forms of vitamin D. The one that's become very popular is to use 25-hydroxyvitamin D itself, calcifidiol. And here we're looking at an effect of vitamin D on the adaptive immune system. So really what we're looking at is vitamin D as a nutrient, and on the other hand, vitamin D as a drug. And unfortunately, I think these two sides to the vitamin D story tend to get mixed up a lot. And so people tend to view this all as being one story, when in reality, we have two stories here. So just to give you one example of some work that I was involved with at the Queen Elizabeth Hospital in Birmingham. This is a study carried out by David Thickett and his colleagues in the intensive care unit in the QE. And what they were looking at was actually the effect of vitamin D levels on the development of COVID-19 in UK healthcare workers. And these are people who are thought to be at risk of getting COVID-19 infection. And what they found was that seroconversion by COVID-19 was indeed higher in healthcare workers with vitamin D deficiencies. So these are not people who've necessarily gone on to go into hospital, but they've just become positive for COVID-19. And this was more prominent in those people who are vitamin D deficient. And the seroconversion was also high in BAME or ethnic groups, particularly in male populations who are vitamin D deficient. And this is important because the study, and I've just highlighted this in red here, shows that within ethnic communities within the West Midlands area where the study was carried out, 43% of people were vitamin D deficient in these populations compared to the white population. So it's quite a big difference. And we're talking about people who are less than 30 nanomolar 25D. So it's very low levels indeed. So this is vitamin D in the setting of community or public health. But what about vitamin D in the setting of a hospital, of somebody who's already become infected? So here I have a rather complex, busy picture, but I think it's also a very, very beautiful image of the different drugs that have been designed for use of treatment of COVID-19 for hospitalized patients. So this is produced by my colleagues here at the University of Birmingham, and it just highlights in purple boxes the different drugs that have been designed for different places within the coronavirus replication and immune response pathway. So here we have, of course, that the coronavirus can interact with the ACE2 receptor. That may be a target for drugs. Viral replication is another potential target. The induction of complement activation is another target. And then we have, of course, activation of the immune system, the innate immune system, neutrophils, macrophages, and so on. And then the development of an adaptive immune response through the induction of various inflammatory cytokines, such as interleukin-6, TNF-alpha, and so on. And what we can see here is I've overlaid onto this picture the areas in which vitamin D could also have a role in interacting with this viral pathway. So vitamin D is known to regulate ACE2 receptor expression. It can also help with the barrier integrity that could improve the consequences of inflammation occurring after development of COVID-19. So one of the reasons you may die eventually if you have COVID-19 is to development of pulmonary edema, the breakdown of the endothelial barriers within the lungs, and the development of acute respiratory distress syndrome. We know that vitamin D is very good at promoting barrier integrity, and this has been shown within the GI tract. There are also some interesting studies coming through suggesting that there's something very similar happens within the lungs. But primarily, the major action of vitamin D is to be very much like the other steroids that are widely used for treatment of COVID-19 patients, such as dexamethasone, for example, or cortisol. So vitamin D acts in the same way. It's a steroid hormone. It acts as an anti-inflammatory agent. But like all of the other steroid hormones here, if you're using vitamin D, you have to use either a very high dose or a different form of vitamin D. And this is the approach that's been taken by several groups now, which is to skip using good old vitamin D that we can get from our local supermarket or health food store, and instead use 25-hydroxyvitamin D itself, calcifediol. And there's a good reason for this. This is a study carried out by my colleagues at UCLA, John Adams and his team, where he looked at the response, the induction of serum 25D levels in patients who received just plain old vitamin D, vitamin D3, which is at 2,400 international units per day, or they were given 20 micrograms per day of calcifediol per day. And what you can see is the solid line here is the levels of 25D went up much more quickly and were retained at a high level, maintained at a high level, if you use calcifediol rather than just general vitamin D. And that's really important because it means that you're able to use this form of vitamin D in a more acute setting. And this was the reason why calcifediol was used in this particular study here from Spain, which also included Roger Bouillon from Leuven. This was published as a preprint on the Lancet site recently, and it got an awful lot of attention. But it has actually been taken down from the website now, and partly because the study, although it was hugely successful in terms of decreasing disease severity and mortality, there was some issues in relation to the randomization of patients within this trial. So I think it'll be interesting to see whether that comes back in some sort of form, but it certainly gave the idea that you could use a particular form of vitamin D in an acute anti-inflammatory setting. However, a week later, there was another study published which also used short-term treatment with vitamin D, and this didn't work. This was published in JAMA just a couple of weeks ago, and it showed that if you have a single dose of just vitamin D, this is not calcifediol, this is plain old vitamin D, but you give it a very high dose, 200,000 international units, and then look for effects over a relatively short space of time. In this case, it was only about 7 days. There was no effect. And I think it's really important to point this out. We know from the previous study that Adrian Martineau published that these bolus doses just don't work very well. And in this particular case, it was just too short a period of time, I think, to really see anything. But nevertheless, in interest of balance, it was a negative study. So that really brings you right up to date. These are studies that have been carried out really in the last, published in the last couple of weeks. So where do we go from here with vitamin D? I think obviously the question which is always asked about vitamin D is, do we need more supplementation trials? There are trials that are currently underway. I think that's fair to say. So we'll find out more in the next few months about whether vitamin D supplementation has an impact in the community, but also in the setting of hospitals in acute care. I think one of the problems here, as we've seen from both of the last studies that I talked about, was that the trials in both cases were flawed in their design. And it's very difficult, I think, for these trials to be perfect under the current setup. But I think that it is important to recognize that there are certain ways in which vitamin D trials are going to work and other ways which they're almost designed to fail. And the main factor really that fits into all of this is that we don't know what the optimal level is for vitamin D to improve immune function. We know that if you get over 30 nanomoles per liter 25D, you're probably going to prevent rickets and certainly do some benefit for your bones. But what is the optimal level for improved optimal immune function? We just don't know this. And that's something I think is really important for us to understand going forward. Point 3, I don't believe that we're measuring the right form of vitamin D. At the very center here, we have 25D, and this is always used in every single study of vitamin D as representing vitamin D biological function. And yet it's an inactive form of vitamin D that circulates bound very tightly to a globulin called vitamin D binding protein, which in our studies really prevents the 25D getting into tissues like immune cells. And so there is some suggestion from groups like ourselves that it's actually the tiny amount of 25D that's unbound, that's free, that is actually driving immune responses. So maybe we should be looking at free 25D. But remember, this is not even the active form of vitamin D. This down here in red, 125D is the active form of vitamin D. Should we be measuring this as well when we look at responses to vitamin D? We've got to bear in mind, actually, this 125D form of vitamin D is very tightly regulated by feedback control by an enzyme called CYP24A1, which triggers all these catabolic forms of vitamin D. And there are a number of researchers now using ratios of 25D to 2425D or 125D to 2425D to give some better indication of the biological potential of vitamin D. But even if we put that to one side, there are other sides to vitamin D that haven't really been looked at yet. If we just give somebody vitamin D supplementation or they go in the sun, they produce vitamin D. And this can be metabolized by the enzyme CYP11A1. This is the side chain cleavage enzyme, one of the most important enzymes in steroidogenesis. And it drives the production of other forms of vitamin D that are totally different from 25D and 125D. Things like 17-hydroxyvitamin D, 20-hydroxyvitamin D. These are metabolites that can bind to other nuclear receptors. They don't have to bind to the vitamin D receptor, but they have completely different biology. And finally, some work that we're doing at the minute, and this is being carried out primarily by my postdoc, Carl Jenkinson, is looking at conjugated forms of 25D. If we look within the circulation, actually, we think 25D is the most abundant form of vitamin D in the circulation, but in reality, it's probably 25D sulfate. There's at least as much as 25D there. And 25D sulfate is circulating around in large amounts. It cannot access target cells the same way the 25D does. And so here, the access to cells involves channels that take up sulfate and glucuronide forms of 25D. But these still could be very important in some tissues having a good biological function. We just never studied these yet, but I think it will be important to do that in future. And finally, one point I'd like to make is that I'd like to go back to the very beginning of why we took an interest in vitamin D in the immune system in the first place. And this was because of some fundamental understanding of the mechanisms involved in vitamin D responses. And I think we need to go back and revisit these. As Chris pointed out at the very start, we had a hugely successful joint PhD project over the last 4 years, which has driven a new concept for vitamin D and its immune response. And this is the idea that vitamin D drives a lot of effects through immunometabolism. And I'll just sort of finish with this particular image here. This is hopefully the graphical abstract for a paper that Amadeo has attempted to get published. I'm hoping that it'll be accepted for publication soon. But the whole idea is that vitamin D acting within immune cells drives glucose metabolism, electron transport, the TCA cycle. And in doing so, it produces increased levels of fatty acid synthesis, which are really essential for the tolerogenic anti-inflammatory effects of vitamin D. So I think going forward, I'm hoping that our collaboration with the guys in Maastricth will help to expand our understanding of the basic mechanisms about how vitamin D interacts with the immune system. And hopefully we'll then be able to apply this to have a better understanding of how vitamin D may have benefits in terms of diseases such as COVID-19. Okay, I'm going to finish there, except to thank again Amadeo for producing the data from this collaboration with Maastricth, Chris and Susan Court for a fabulous 4 years of interaction with them and Maastricht University, Carl Jenkinson generating all of the vitamin D metabolite data and having a great time on his Marie Curie fellowship in Sydney, and then my collaborators at the University of Birmingham and UCLA, as well as sponsors for this work. Thank you for your attention. So first question I can see is meta-analysis is only hypothesis finding exercise. Do you know? Yeah, I think that's exactly true, Jolanta. And I think there is one study. Obviously, the individual studies that were carried out to make up the meta-analysis were addressed to trying to look at the effects of vitamin D supplementation on respiratory infections. But there is a one underway in the UK at the moment, which is looking at specifically at vitamin D supplementation and how it will affect COVID-19 infection. In fact, I think that will look at all acute respiratory infections as well. So obviously this involves a lot of people. I think it's one of the issues with all these trials is that, particularly with COVID-19, it's still relatively rare to get the disease. So it's difficult to carry out these sort of supplementation study, and I'm pretty sure we'll see a lot more over the coming year. In terms of your other question about inflammatory bowel disease, yeah, I was involved with one of these, but it was actually stopped because of the COVID-19 crisis, and we weren't able to complete this trial. I think there are some other studies that are underway looking at vitamin D in the setting of inflammatory supplementation in inflammatory bowel disease. So yes, I think this is another area that's of some interest as well. So second question. Okay, I'll just complete the questions that are in the chat box. So the disease pathways are now more or less a summary. Okay, I'm not sure what that question is actually.
No, I think you can just do the ones in Q&A, Martin.
Okay.
Because they were all copied there.
Okay, what happens if a person takes excess vitamin D supplements? Good question. Certainly the UK government, they're very, very much preoccupied with this. They believe this is a problem. But in reality, there have been really very few cases of vitamin D overdosing have occurred. UK government says there is no chance that you will ever get any problems if you take less than 4,000 international units per day. If you do decide to eat all of the jar of vitamin D supplements, there's a risk that you could become hypercalcemic. That will make you quite sick and also increases risk of getting things such as renal stones. But really, I think it's a little bit overplayed. That's my own personal opinion, that the risk of vitamin D toxicity is always mentioned, quite rightly, but there have been so very few cases of this. Vidisha says, it has been shown that even in the sunny south, that's Greece, Italy, and Spain, people are actually deficient in vitamin D. And okay, yes, that's also true of the sunniest countries in the world. If you look at Saudi Arabia, it's also one of the countries with the highest levels of vitamin D deficiency. And as you say, quite rightly point out, that's because people limit their exposure to the sun, either through dress codes or what they wear, just covering up, or as has been experienced in places like Australia, sunscreen will effectively block out all of the UVB light that's required to generate the vitamin D within the skin. So sunscreen, yeah, I mean, we should all use that. There is apparently, I believe, some advice in Australia now to people that, they should spend 10 minutes on the beach first before they put sunscreen on to allow themselves to get a little bit of exposure to make vitamin D. I believe that's the case. So,
Can I quickly link that to something else you said? I remember reading a paper, I forgot which one, that indicated that there might actually be genetic variants in the metabolism that then affect the biomarker levels more than the active forms, which relates to something you taught.
Yes, that's absolutely true. I mean, the vitamin D system is one that's been well studied genetically in terms of GWAS analysis. And there are various genes associated with vitamin D metabolism, but particularly the vitamin D binding protein, which shuttles vitamin D around the body, helps for it to be recycled via the kidney, that have been shown to contribute to a genetic risk of vitamin D deficiency or sufficiency. So depending on your various SNPs that you may have, you may be genetically predisposed to have higher or lower levels of vitamin D. But, and this is crucially important because a lot of studies are then, of course, using Mendelian randomization to say, hey, what's your lifetime exposure to low vitamin D? That's surely more important. The fact of the matter is that that genetic component of your vitamin D status is still really low, maybe at best about 7.5%, possibly less, 5%. So, you know, you go and spend one day at the beach, you're probably going to outstrip any difference that may occur in terms of your genetics. But it is a factor, and some people are at greater risk of vitamin D deficiency than others because of their genetic background. So I don't know if that answers your question, Chris. But Ralph Stefan says, is there a long-term vitamin D storage, either longer than a month, that is filled in summer? Yeah, I mean, essentially, your 25 hydroxyvitamin D levels have got a half-life rather of several weeks. There is some storage of 25 hydroxyvitamin D and vitamin D within adipose tissue. So this is one of the arguments that people who have high amounts of adipose tissue, who have high BMI, for example, may be at greater risk of vitamin D, 25 hydroxyvitamin D deficiency, because they have more of their 25D within adipose tissue. But the idea is that as we leave summer, go into autumn, we start to deplete those stocks that are in adipose tissue, so that by the time you reach, like now, this very time of the year, we no longer have any longer term storage of 25D within adipose tissue. And the adipose stores are also thought to be potentially really acting almost like a sponge and limiting the amount of 25D that's available for other tissues such as the immune system. So this is one of the rationale for often linking BMI with poor vitamin D levels. So yeah, it's an issue, I think. Anonymous 10D, is there a role of calcium in the activation of the immune system effect of vitamin D? Yeah, you know, I don't really know. I mean, I certainly most of the supplementation studies that have been done looking at immune effects have not looked at combined calcium, for example, but many of the other studies have, and they tend to be complex. I think if you're using a combined calcium supplementation, because calcium will regulate parathyroid hormone, and that's one of the key promoters of vitamin D activation. So you get a very mixed message there. Now, if you go to the basic immune response to vitamin D, I'm not sure. I think that's a good question, and they could well be in some areas. I mentioned in one of my slides how intracellular calcium levels may be important for regulation of antiviral responses. So there's a number of people are suggesting that maybe vitamin D does help to promote intracellular calcium levels in some immune cells, and that could have a positive effect in regulating viral infection. I'm not really sure is the answer, but I think that's a possibility. Okay, is daily supplementation more effective than weekly in increasing? You know, if you talk to anybody in the vitamin D field, they would say, yeah, it's better if you take a small amount of vitamin D every day rather than a weekly dose. But that weekly dose can be much more relevant in clinical settings. So it's a weekly dose is often given to people who are severely vitamin D deficient, maybe a child that's at risk, severe risk at rickets. They'll give them weekly because it's bigger dose and it will get them up to a healthy level very quickly. But then often they then go on to a maintenance dose. So I would take a daily dose, but it's yeah. But weekly is better than monthly. Monthly bolus dosing doesn't appear to work very well at all. So on what basis? Oh, sorry, I've got all these. On what basis is the level of vitamin D that indicates deficiency derived? That's another great question. Look, I could spend the whole day talking about the levels of vitamin D that are thought to be important. The UK government has decided that less than 30 nanomoles per litre is bad for rickets. That doesn't mean to say if you're above that you're doing particularly well. The Institute of Medicine in North America says, you should be above 50 nanomoles per litre for your good bone health. That's not just rickets, that's osteoporosis and so on. And then the American Endocrine Society said even that's not good enough. They recommend people getting to 75 nanomoles per litre to have good musculoskeletal health. It's a contentious issue, and I'm not sure. It depends on what you hope to achieve. UK government said we've set a low level below which we'd rather you didn't go, because that's something that you can tackle quite well on a population level. If you just tell everybody, try and stay above this low level, you're more likely to be successful. Anyway, you mentioned that large dose 1 monthly treatment is not advisable, but then the calcifediol is beneficial. Yes. It's primarily a once a month treatment. Well, actually, yes. So the idea is that giving a big bolus dose of vitamin D doesn't appear to work, and the rationale for that is that you may be triggering a lot of feedback control enzymes by giving a very big dose. So essentially, you're flooding the system with vitamin D, then flooding with 25D, which then it triggers a lot of feedback control mechanisms. Now, you used to say, well, if I give a lot of 25D as calcifediol, I'm just doing the same thing. You probably are, but you're getting more of the 25D into the system to affect the immune cells compared to vitamin D. There's a lag with the vitamin D. It's got to go to the liver, get converted to 25D. There's a problem. There's inherent inefficiency there, but then you've got to wait for that 25D level to come up and affect the immune cells. So purely a calcifediol, it's a sort of benefit versus risk. You're triggering feedback mechanisms, but you're also getting the 25D to the immune cells to have a positive effect quicker and more efficiently. Okay, this is George Leitz. During COVID-19, we expect to see that local stores of vitamin A are reduced in clinically ill patients, and that this may affect long COVID. Yeah, there are some new projects coming out. Exactly, I think there's a lot of interest now in people who've had COVID, have survived, and are recuperating, and whether or not vitamin D outside of those immune effects I described, the innate or adaptive immune system, does vitamin D have a broader impact on improving musculoskeletal health in recovering COVID patients? That's another trial that I'm involved with our collaborators at the University of Nottingham who are interested in exactly this. I think there'll be new information on this coming through fairly soon. Meta-analysis, okay, well, I think I already mentioned that. Any risk of vitamin D overdosing? Okay. Would you think that in COVID-19 we need to look at 125D and also 2425D? Yeah, I think so. I think with all of these immune diseases, I think we've been too reliant for too long on measuring one form of vitamin D and saying that it's a surrogate measure of the impact of vitamin D on diverse effects of human health. I don't think that we can. It's very convenient. It's really easy to measure 25-hydroxyvitamin D. You take a tiny bit of filter from a blood spot from a newborn child. You can measure the 25D in that. It's fine, but it gives you a very limited picture of the biological activity of vitamin D in that individual. So yes, I would agree with that. We need to do more of that. That's Carl, my postdoc working in Sydney. His Marie Curie fellowship is to do precisely this, to design more high throughput effective assays that look at more vitamin D metabolites and give us more information. This happens in all the other steroid hormones. If you look at androgens or glucocorticoids, they don't just rely on cortisol or testosterone and that's it. That's what we do with vitamin D, you know. Anyway, can you please comment on individual variability in vitamin D metabolism? Yeah, FOC1 is obviously a prominent SNP within the vitamin D receptor gene. Yeah, okay, I could have shown you a beautiful picture which was given to me by Neil Binkley, where he took 20 patients and he gave them all the same level of vitamin D supplementation and then measured their 25D levels a month later. And the variability in the effect was astonishing. Some people went up enormously, others hardly at all. Some people started off low and went up really high. Others started off high and didn't go up much at all. So very variable. And that's another one of the reasons why there's a lot of interest in using calcifediol in many settings, because you skip out that initial step, converting vitamin D to 25D appears to be inherently variable. And in addition to that, you point out the actual response to vitamin D through vitamin D receptor polymorphisms is also there as well. So, you know, it's not a simple system. Okay.
Martin,
yeah.
It's about 3 minutes to the hour, and
yeah,
a lot of people will probably have to leave early, so I would want to break in. First of all, thank you on behalf of the almost 200 people. We're over 200, some are leaving already, that were here. And I see a lot of comments also in the chat of people that tell you it was a great presentation. We did a quick poll. I would want to summarize that briefly. Almost equal numbers of you said no, we're not interested, not taking too much care of vitamin D, or telling me I'm either going out more or taking supplements, which means, and in total that's still 3/4, and over half of you are taking supplements. Also about half of you said that they are now more attentive to vitamin D because of the COVID-19 crisis. Although a small majority of that again said that is mostly because I have to stay home all the time and I realize my vitamin D might actually get low, right? So it's not only because of risk of infection. And then a striking 98% of you, and I noticed that 85% of the people voted, said, yes, we should inform the public more.
That's good.
Yeah, we could actually use that in that information, I guess.
I think that's important, Chris. And I mean, as you said, leaving aside the current pandemic, I think that the idea that vitamin D deficiency is very prevalent, affects other aspects of our health, I think is important, and people at least aware that there's a risk. I think that's, they have a choice then, and I think governments are beginning to understand this a little bit more. So
I think we're going to end it here then, Martin. Once again, thank you very much.
My pleasure.
All right. Thank you all very much.
Okay, thank you.
Thank you, everyone.
Bye, everyone.